Good afternoon, everybody. It's a pleasure to be here. I believe that quantum computing is ready to branch out into areas such as healthcare, and I believe that this is a sign of maturation of quantum computing as a field. And my invitation to a conference, distinguished conference such as this, I feel is a proof of such maturation, because you see, I'm not a quantum physicist or a computer scientist. I obtained my PhD in the Weizmann Institute of Science in immunology, and then went on to pursue a postdoctoral fellowship at Keio University here in Japan in microbiology. And I would have continued upon the traditional path of biomedical career if it wasn't for the establishment of Microbiome Quantum Research Center at Keio University, with a mission of achieving fusion between quantum computing and healthcare. At the Bio2Q Quantum Research Center, we believe that when it comes to applying quantum computing to healthcare challenges, there is this additional bottleneck. In addition to developing hardware, in addition to developing quantum algorithms, this bottleneck is really in translating biological problems to the form that is accessible to quantum computing. What I'm talking about here is that the fact that this audience, of course, knows that all classical computation is a subset of quantum computation, and from this follows that all classical bioinformatics is a subset of quantum bioinformatics. But we then need to expand this area of quantum bioinformatics, and to do so, we need to find novel problems to formulate in a way that is accessible for quantum computers. And in today's talk, I will present such an example. In the long name of our center, there are also these two words that human microbiome. And what is microbiome? Microbiome refers to a community of microorganisms that are associated with the human body and that affect human health. Human health and microorganisms have a long history. That started in the late 19th century when Robert Koch discovered that infectious diseases such as bubonic plague are caused by bacteria. And later on, when Alexander Fleming discovered the first antibiotic, we learned how to defeat those dangerous bad bacteria that cause diseases. And that has led to the first leap in expanding human lifespan. And these fundamental discoveries formed the notion that bad bacteria cause diseases. But since then, the paradigm of our understanding of the relationship between microorganisms and human health has really changed dramatically. We now appreciate that actually our health to a large degree depends on good bacteria, and that the definition of health right now includes healthy microbiome as well. And a particular strong example of this has been demonstrated a few years ago in the area of cancer. So we have found that actually some cancer immunotherapy simply fails if you co-administer it together with broad-spectrum antibiotics. So this means that there are some good bacteria that are required for cancer therapy to work. So altogether, our paradigm in the 21st century is that there are a lot of good bacteria that are required for our health, and in the area of cancer, that there are some good bacteria that help treat cancer. When we talk about microbiome, what are the dimensions of data that we deal with? So just in one slide, there are on the order of 10 to the 6 different kinds of bacteria associated with the human body. In terms of numbers, if you just count the cells, human cells and bacterial cells, the ratio is 1:1. But when it comes to chemical reactions that our cells and bacterial cells can carry out, our bacterial symbionts have us beaten completely, because they are able to carry out almost 2 orders of magnitude more chemical reactions than our own cells can. And that is the basis for our reliance on bacteria for our health, because our cells, human cells, basically outsource a lot of chemical reactions that are required to support our health to our bacterial symbionts. And this is the reason why only 10% of our human diseases are actually caused by bad bacteria, while 90% of our diseases are associated with the loss of good bacteria. And why am I talking about this in a quantum conference? That is because this is a lot of combinatorial complexity here in the human microbiome, and that I mentioned that in the previous slide that we believe that the next leap in expanding human lifespan will come if we are able to understand how these 10 to the 6 different types of bacteria interact in complicated ways to support human health. So what is the particular problem in the space of human microbiome and human health that we think can be solved with quantum computing? This problem has to do with antibiotics again. Since their invention in the beginning of the 20th century, we really abused this miraculous drug. And in particular, we overused them in hospitals. And this overuse has led to the evolution of bacteria that are resistant to almost all of antibiotics that we have at our disposal. And in a vicious cycle, hospitals are the places where, with the high concentration of drug-resistant bacteria that are dangerous, but also hospitals are where our most vulnerable like members of human population are also concentrated. And this, of course, is a recipe for disaster, and this disaster is slowly unfolding, and both in terms of loss of human life and in terms of financial burden on our health systems globally. So in 2019, 1.3 million people lost their lives due to hospital-acquired multi-drug resistant infections, and the financial burden on global healthcare systems reached 66 billion per year. And because we are continuing to overuse antibiotics, more drug-resistant infections are rising every year, and but our best estimates suggest that if we don't develop an alternative solution, then by 2050, we'll be losing close to 10 million people every year. And of course, the financial burden will be even greater than now. So we really need an alternative solution. So if we cannot kill this bacteria, these infections with drugs, what can we do? And when all else fails, go back to fundamentals. And bacteria, no matter how resistant they are to drugs, are still living systems, which means they require food to grow. And if we deprive them of this food, we starve them, then they cannot grow, How can we deprive drug-resistant infections from food? So if we know, let's say we have a target, drug-resistant pathogen or bacteria, and we know which nutrients it needs, it's highlighted here in red. So then we can find like good bacteria that are not dangerous or even perhaps beneficial bacteria that consume the exact same nutrients that are required by the bad bacteria, by the pathogen. And then we can administer them in a pill to an infected patient. This good bacteria will enter into competition with the bad bacteria, starving it and killing it. This is a pretty concept. But we were able to actually prove it last year in a study published in Nature. Here is the representative data, real data, not a simulation, real experimental data. On the y-axis here, we have level of infection by one of the deadliest drug-resistant bacteria globally, Klebsiella pneumoniae. And on the x-axis, we have time. And there are 3 experimental groups, 2 controls, and in red we have a treatment group, and you can appreciate that a single dose of a combination of good bacteria that consume the nutrients required by the drug, otherwise very difficult to treat pathogen, leads to a dramatic drop by 4 orders of magnitude in terms of level of infection to a level that is no longer dangerous. So this suggests that combinations of this good bacteria can be a solution to this looming and growing global medical challenge. However, there are several bottlenecks in terms of scaling of this technology. One is time. It took us 7 years of trial and error to find this effective combination of good bacteria to defeat that drug-resistant infection, while on average, from the moment we introduce a new antibiotic into clinic, in 3 years, a drug-resistant pathogen emerges and starts killing people in hospitals. So we really need a much faster way to develop such therapeutics. Second bottleneck is combinatorial complexity. I mentioned that there are on the order of 10 to the power of 6 of different types of bacteria associated with the human body. And of course, the number of combinations is a factorially scaling function. So there is no way we can efficiently explore this combinatorial space experimentally, which is the state-of-the-art method right now. So we need alternative approaches. So what is the solution to these bottlenecks? The solution is to reinterpret the task of finding an effective combination as a combinatorial optimization problem. So what we have is a target drug-resistant bacteria, and the given data is the nutrient utilization, which nutrients this bacteria requires. And we have the same data for good bacteria as well. And then the task of identifying an effective therapeutic would be, is equivalent to finding a minimal set of good bacteria that together sum to equal or greater than the vector of the pathogen. And to achieve this, we have developed a hybrid classical quantum workflow where we have input data. Input data, as I mentioned, is a dimension of 10 to the 6 by 10 to the 6 with a sparsity of 19%. So it's a fairly dense matrix. So then, of course, we have to do pre-processing classically, formulating a Q cost function, which we then optimize using several quantum platforms, quantum annealing platforms, as well as gate-based quantum annealing platforms. And the output is the kind of the therapeutic combinations which we can rapidly test in the lab and validate. So this approach offers several significant advantages over current state of the art. So this technology of like live therapeutics, so good bacteria in a pill, is actually being right now commercialized by several pharmaceutical players. But to the best of our knowledge, all the products on the market right now are designed empirically. So there's no even classical algorithmic solution for this. So the first major advantage is, of course, time. So we, in our previous study last year, it took us 7 years to identify good combinations, and now we can do it in hours. Second advantage is in optimization of cost effectiveness. So in that nature study I just mentioned, the best combination we found was of the size 18. But using the exact same data from the same project and using our novel computational workflow, we were able to identify a combination of the size 10 with the same efficiency. So dramatically, almost half, or reducing by half the complexity of the therapeutical combination, thereby improving the cost effectiveness of solution. And the second use case is in prediction of susceptibility to infection. As I mentioned, statistically, when 13 people walk into a hospital, one will get infected by a drug-resistant pathogen, but at the moment, there is no way to tell which person is the most susceptible and needs most care. So as we enter the age of precision medicine and personalized medicine, using the data on the composition of microbiome of a patient and using our algorithm, we'll be able to simulate the competition between the pathogen and the native microbiome of the patient and predict who is the most susceptible person. And one way this information can be then acted upon is, for example, by administering the therapeutic combination preventatively rather than after the infection has set root and caused damage. And we have validated the second use case as well on a cohort of real patients in the Keio University Hospital. I mentioned in the very beginning of the talk that we believe that understanding of human microbiome will lead to the next leap in human longevity. So in that framework, in the study, we focused on a cohort of centenarians, so people with an average age of over 100. And we also had 2 control groups, healthy older people and young adults. And we took 10 most deadly pathogens that are together responsible for over 70% of drug-resistant deaths globally, and simulated competition between them and the microbiomes of these groups of patients. And we found something interesting. We found that young people enjoy ecological protection from invasion by these pathogens, and this protection is retained by centenarians, but is lost by people who don't get to live to such an advanced age. So this suggests that another use for combinations of good bacteria in a pill. Like in the first use case, we utilized it to treat already occurred infections. But this result suggests that we can also use them to prevent infection from occurring in the first place in a personalized manner. So to extend the umbrella of protection from that is naturally enjoyed by centenarians onto a regular mortal people. With that, I would like to thank you for your attention. If you are a quantum computing hardware manufacturer or a software developer, and you're interested in a demonstration of how your technology can help solve problems in the biomedical sphere, the cutting edge of science, please come talk to us. We have, in addition to these stories, we have other completely novel problem formulations that we are working upon, and we'll be happy to talk to you. And finally, if you are a postdoc, we have open positions, so please feel free to contact us. And the last thing is I'm also a member of SQAI. SQAI is a proud sponsor of Q2B conference. We have a booth. So if you're interested in great research that my colleagues are doing, please come talk to us. Thank you for your attention.
We do have time for a few questions, so if anyone has questions for Timur.
I know there's a cocktail hour. There's just one lecture after mine, so we can definitely discuss any questions you have or proposals at the cocktail hour. Oh, please.
Yeah, actually, I'm not so much familiar with such microbiome actually, but my question is, could you comment at the is that the causal relationship between your good microbiome to your pathogens, or it's just statistical correlations or something like that?
Thank you for a great question, and it's right to the point. So we come. This whole research study started from a completely biological project in which we proved in a causal way that you can have a very high level of infection, then administer the combination of good bacteria that completely drives down the level of infection by 4 orders of magnitude. So it's a completely causal relationship. But in that, over the course of that project, we encountered combinatorial barrier that prevents us from developing such therapeutics in an efficient way at high scale. So that was the motivation to then develop algorithmic solutions to overcome those bottlenecks and limitations.
Thank you very much.
Thank you.